Dear colleagues, I'm very happy that the order was like it was, that the previous speaker ended up with butterflies. So I'm coming to the butterflies also, but from a little bit different point of view. So we have a project that does not deal with this climate change in a very direct way, rather opposite. We have a problem to resolve to define the biodiversity. And this project is called BIOTAGENE, which means that we are dealing with genes, actually, the genes from the soil. And I represent the Tallinn University of Technology. I am the chairman of the Center for Biology of Integrated Systems. And the other part is the University of Tartu, the Institute of Ecology and Earth Sciences. And we have approximately 1.6 million euros for this project altogether. And the policy area is innovation. So what's and where is this project taking place? So we do a practical work. The project is run only in Estonia. However, it includes also the sampling from all countries around the Baltic Sea. It's a good case because we are not in India or in Brazil, where picking soil is complex because you could be just arrested and jailed, even if in Poland, in Pelavesia, you cannot actually take soil samples without prior agreement with some authorities. So we're going to sample some soil, and why we're doing this, we have the objective to develop and demonstrate a novel approach for environmental monitoring based on the metagenome data. So that means that we are analyzing the DNA in the soil, and in the soil there is a lot of life. There is much more life than usually people expect. What is the case is that the green biologists haven't really realized that there is so much life. Geologists, however, 5 years ago already found it. So when I tell today to an average green biologist there is approximately 12,000 taxons of bacteria in the soil, in 1 gram of soil, they usually don't believe it. However, that's the truth. And the total genome of 1 gram of soil from different species is slightly bigger than the human genome. So we make this new method, and we use our previous information we have, and then we prepare a kit, a prototype of a kit that everybody could use. So now the question, can we access the biodiversity from the DNA point of view, and why it's better than butterflies? So until now, and even now in the previous project, it's very good to use these microorganisms. It's simple because it's very easy to use. The problem is that you cannot use the same methodology in Finland and in Sicily, because the butterflies are different and the birds are different and the plants are different. So we have to have something that is common, that could be used wherever, on Spitsbergen or in Sicily, and even actually, since the climate change is not a local European problem but a global one, maybe in Panama or in Argentina. So the entire idea to go for this biodiversity from a DNA point of view came from the fact that some groups of microorganisms in the soil, particularly some symbiotic fungi, are have the same kind of biodiversity and the same species in Estonia as in a forest that has been untouched by human hand for 200 years. The same species are found in Panama and in French Guiana, for example. So what we do is that we assume that the environmental DNA has imprint, and looking for that one, we can tell what is the situation. And the next question is how our action, what we do as the biggest parasites in the nature, what humans are, how we affect the other populations around us. So we took in the case study 4 different places: the primeval forest as a template for intact ecosystem. We took also successional natural forest on the heavy recreational pressure. We took the oilseed rape field, and we took a place in Estonia which is a partially vegetated industrial waste as an extremely disturbed ecosystem. And then when we analyze the taxon set of these places with different methods. We used three methods. We used a multidimensional scaling with very short distance measure. We use a spectral analysis, so-called Laplacian eigenmap, and we use a GLMNet package to see the general linear models for the convex penalties. We came to this conclusion that I would like to demonstrate to you, that if we start from the green, that is a real forest, untouched, and we go to the mining, which is red up there, then we see a very, very clear vector of species distribution between these places with different human involvement in the disturbing of the nature. So it's not, however, not the entire truth. Because if you treat this kind of data and you want to predict how to use this data in the future for the wider evaluation, please pay attention to the left side. This is a spectral analysis using Laplacian eigenmap, where the black dots, which are upper right there, present the distribution of different bacterial taxons in the untouched forest. There we see that the distribution of different species is very much the same. They are clustering all together. That means that all 12,000 taxons we have analyzed are located the same way in different sampling points. However, if you look like the other places, like in the field or in the wasteland, specifically in the recreational forest, the distribution is much wider, which means that the human influence on the soil microorganism community is really very big. Now, the third biggest question, that if we do this analysis and we create the prototype, is there a possibility to select the markers that can be used by other people who want to evaluate the environment? So we use this general linear model with convex penalties, and we show that in our model case, only 9 taxons are sufficient to analyze. That means that we can make a relatively simple method, a relatively simple kit to analyze into which category of environment the particular one goes, or how big is the human influence, how bad is the case. And we have also shown that with a larger data sets, we need only maximally 35 taxons. That means that there's a very little, actually, a very little amount of experimental work to be done to define how big is the human influence on the environment. So, and that makes a possibility that we can make a relatively very simple kit, or actually in the future we think about the device, kind of not a bedside, but a field side equipment that you can go out and test how bad is the situation in a particular place. If it is a park in the city, or it's a protected area, or it's a field, how much it is affected by our human action. So that's a very short project. I won't tell you all these things that we of course gonna produce all these leaflets and DVDs and web pages and whatever else that have to be produced in the live project. But however, we would like to create a real output, something physical and which is possible to touch with hands and to use to measure the human influence on the nature. Thank you.